We have been undertaking an environmental DNA water sampling project in the Wollombi catchment with the local community to try and see if we can detect the presence of platypus and rakali. These two species are our focus because they represent apex species in the riparian environment. If they're about, that indicates that there is good habitat and a functioning ecosystem.
Platypus and the rakali are native semi-aquatic mammals, so they spend part of their time swimming around in the water foraging for food, especially at night. They're carnivorous species, so they feed mainly on things like insects and crustaceans, yabbies, shrimps, and then they also make burrows in the stream banks where they'll shelter and raise their young.
The Wollombi Valley it's a large catchment and it's quite unique in that it is a sand-based system. It's surrounded by sandstone country and it is just a very picturesque valley. The catchment has experienced some very extreme weather and climate events, from the droughts and bushfires in 2019-2020, and then following through with a number of flood events which culminated in a huge flood in July 2022. The result of these climatic effects have been that the habitat within the riparian channel has changed significantly. So these animals that live in deep refuge pools, these pools have changed. You've had some pools that have filled up with sediment from the flooding events and other areas where we've had massive erosion. So because there's been such a significant shift in habitat, we were fortunate to get some funding from the New South Wales Environmental Trust to undertake this project. And we had some fauna surveys undertaken in summer by Dr. Bruce Chessman, focusing on iconic species in the Wollombi catchment.
I did a survey of 13 sites focused on 3 aquatic species, the Hunter River turtle, that's a short-necked native turtle, and the platypus, and the rakali, which is sometimes known as the native water rat. The Hunter River short-necked turtle was very common, large numbers at several of the sites, and a lot of very young ones as well, which is a great sign. The platypus and the rakali were not so common. I was only able to observe platypus at one site, and rakali at 2 of the sites. They're not always that easy to observe where they're present. They may be in their burrows rather than swimming around.
Through our fauna surveys that were undertaken, it was a recommendation to use eDNA as an alternative method because these animals are very cryptic.
It's a way of getting more information on the distribution of those species and detecting them at sites where the standard survey techniques may not pick them up.
Doing citizen science projects like this, it's a fantastic way to get the community involved. And we have around 30 to 40 volunteers out doing the sampling tests throughout the catchment, which is a great result.
To be able to take this DNA sampling will give us a much better idea of just what is here. And the fact of the new technology that can do this is just amazing.
To take an eDNA sample, firstly, we need to draw up some water from the local creek into a large syringe, and this water is pushed through a filter disc. And then we add a little bit of preservative to the filter disc to preserve whatever DNA particles might be present. And then packaging all those samples up and sending them off to a lab in Melbourne for DNA testing. So once the samples have been tested in the lab, we get the results back. If we get positive results for any of the sample sites, it will give us a good reference for good habitat area. Then we can potentially work with landholders to assist in helping enhance and protect these particular sites.
It's particularly important during drought to be able to protect the refuge pools where these species may be sheltering to survive the drought, and to protect the riparian zones with vegetation that stabilises the banks so the banks don't erode and fill in the pools that those species need.
We'd really like to encourage landholders to enhance the riparian habitat, and they can do this by activities such as fencing out cattle or stock from the riverbanks, planting more native trees and shrubs, and addressing weed infestations. Good riparian habitat looks messy. It's really good to have a variety of trees and shrubs along the riparian channel. as well as fallen timber and logs, which is fantastic for animals to rest on.
We really appreciate what's been done to learn more about just what is here, how to best manage it, and bring the community together on a project where we've all got the one focus.
The results from the eDNA samples will be included on an online mapping platform for all the community members to look at so that they can see what results they got from their site as well as all the other sampling sites across the entire catchment.
I really appreciate the opportunity to be involved in this project, and it's a great initiative to understand more about the ecology of the brook and its tributaries and how the native species are faring.